we were talking about the main limitation of fluorescence microscopy. As any other optical technique, it has a resolution that is limited by the wavelength of light, the so-called diffraction limit. Here's an example. What you see here is a baby neuron. It has a cell body and several protrusions, and after a while, one of those protrusions will decide to become an axon and will start growing very fast. And for example, if you would like to know that, or to understand how that neural polarization takes place, you need to go into the molecular mechanisms inside the neuron. And if you, for example, would like to zoom in one of those growth cones, it looks like that. And the image starts to be blurry. And that's not because the microscope is not working well or anything like that. It's just because you are reaching the physical resolution limit of an optical microscope. And so you would like to go beyond that. So this barrier, this diffraction barrier, it's on the range of a few hundreds of nanometers. So here you see more or less the imaging tools that we have. And optical microscopy, and fluorescence microscopy is just one of them, was limited by the wavelength of light to that, a few hundreds of nanometers. But that limit has been broken by super resolution microscopy. That's what we will see today, how that diffraction barrier can be broken. But before that, just a few words to understand what is the diffraction limit. So imagine that you have a point light source, a mathematical point that emits light in all directions. This is an ideal light source. It doesn't exist. But imagine that you would have such an ideal light source. It's infinitely small and it emits light in all directions. And now you will make an image of that light source with a microscope. That means you will take a lens and collect some of that light and use another lens or the same lens to make an image of that source, right? In geometrical optics, you would say, okay, the image is formed on that point. But in real life, the image is not the point. The image actually is formed when all waves coming from that light source converge in phase. That's how the image is formed. It's basically an interference problem. So you have a cone of waves converging in one point in phase, and that produces an image. So that image has a size. That size is determined by the geometry of the optical system and the wavelength of light, typically of about a few hundreds of nanometers in the lateral dimension and 2 or 3 times worse in the axial direction. So you see the problem. You start with a mathematical point, infinitely small, and you end up with something that has a finite size, right? And that finite size is what will limit your resolution in the end of an optical microscope. So this is the so-called diffraction limit. It was believed to, it is actually a law of physics, and it's written in stone in Jena in a memorial for Ernst Abbe, as you can see there. But let's take a look about what are the practical implications of this. So we don't have light sources that are infinitely small. They don't exist. This was just an exercise. But we have molecules. We have fluorescent molecules. And they're very small. They're about 1 nanometer in size. And we can make an image of them in a microscope. And those images would look like that, 200 nanometers. So you see, we start with something that is not infinitely small, but it's about 1 nanometer. And we end up with something that is 200 times larger in 1 dimension. In 2 dimensions, it's the square of that. And in 3 dimensions, of course, the problem gets larger. So we have a size mismatch of 1 in 200. Well, how bad is that? Imagine that you are watching a football match. So a human being has a size of about 1 meter, right? And the football pitch, a size of about 100 meters. So now imagine that you would look at this football match with a resolution of 100 meters. So this is a size mismatch of 1 in 100. It's still half as bad as in an optical microscope. That's what you would see. Nothing. You're lucky if you can tell that it's a football stadium there. But there's no way to tell that there are 2 teams there, that they are playing, that they are segregated for moments, that they are playing together for moments. No way to tell there is a ball even, bounce around. So we would like to solve that and look inside that limit to start seeing the details of things. And that is what a super resolution does. Like in this case, you see, these are microtubules in a neuron, And outside the circle, you see a confocal image. So that's a diffraction limited image. And inside, you see a super resolved image, in this case by Stefani. And you see things get clearer. You can microtubule bundles that were not possible, were not resolved, are not resolved by diffraction limited imaging. You can see them now, right? So you see more. Here you see another example of nuclear pore complexes. This is an example from the lab of Stefan Hell. This is a diffraction limited image of a nucleus membrane. And now if you apply STED, this is what you get. You start seeing the nuclear pores, how proteins are organized in space with high detail. Here you see a comparison. So you see much more. So super resolution microscopy is really a tool for discovery. This is very important. It's a tool for discovery. You can see things that are impossible to visualize otherwise. And this is another great example from the lab of Xiaowei Chuang. When they visualized already 10 years ago, this membrane-associated periodic skeleton in neurons. And this is another, here you see the contrast better in a later work from our lab. Up there you see a confocal image. In this case, it's a spectrin, a protein in the neuron. If you would look at diffraction limited image like that, you would think spectrin is uniformly distributed all over the place. You have no indication that it would be otherwise. It's really uniform, the intensity. But when you apply super resolution, you realize that the protein is not really uniformly distributed. It's organized on a highly periodic structure. But that structure has a period of about 190 nanometers. So when you apply a diffraction limited microscopy, the signal averages out and it looks really uniform. So you can really discover things applying super resolution microscopy. And that is the main reason why these three physicists got the Nobel Prize in Chemistry a few years back. So let's now take a look how this is possible. Okay, that's the idea. The goal of this talk is that you learn the main concepts behind a super resolution microscopy. So, or also called fluorescence nanoscopy. So how is it possible? How do we break the diffraction limit? We had some of it already this morning, and there are two main ways of doing it. All of them are based on switching on and off the fluorescence of molecules, okay? But in different ways, and we will see. The first way of doing it is called coordinate targeted. Why? Because we will decide with our illumination where and when molecules are able to emit fluorescence or are not able to emit fluorescence. When and where we switch molecules on and off. So, and we will do that with light. So imagine that we have some fluorescent labels, schematically shown here like dots. And the distance between these dots is smaller than the diffraction limit, right? So if we now make an image of that, for example, with a scanning microscope, we will take a laser, we will focus it as tightly as possible, and that focus will have the size of the diffraction limit, about a few hundred nanometers, because we are focusing light. There's no way around it. And now we can scan this over the sample, and then we will register a signal that it would look like this. Pretty much uniform, just like in the example we saw in the neuron. So there's no way to tell that there is a structure underneath that. It's pretty uniform. But now, let's imagine that there is some way to switch fluorescence off induced by light. So if we have a mechanism to suppress fluorescence with light, then we can take our laser focus, we can generate another focus with this shape, right? That I will show you later how you can do that, but it's very simple. Remember, to make a focus of light, you need to put all the waves in phase to get a maximum of intensity. Well, there are ways to manipulate the phase of a beam so that you get in the center destructive interference instead of constructive interference. And in this case, you get a central zero of intensity, but of course, half a wavelength away, you get a maximum. So you can generate this kind of donut-shaped focus with a central zero or a central minimum of intensity. Again, we need some process to switch fluorescence off induced by light. And now we will do that with this focus. So now we superimpose the two. And now the blue laser will excite fluorescence, and the red laser will suppress fluorescence, right? And you already can imagine what will happen. When we scan this over our sample, now the region of space from which we will detect fluorescence became smaller because we are suppressing fluorescence from the periphery. And if that process is saturable, if you can do this more dramatically, then you can reduce, you can shrink and shrink more and more the region of space from which you register fluorescence. And eventually, you can start resolving where the fluorescent labels were. Okay? So that's the first concept of coordinate targeted fluorescence nanoscopy. And based on this concept, there are many methods. All of them work in the same way. The difference is the off-switching mechanism. The difference is the light-driven off-switching mechanism of fluorescence. So there are many ways to switch off fluorescence using light. One of them is stimulated emission. That's rather universal in principle, and one of the first methods developed is called STED. But you can also do it by depleting the ground state. That's called GSD, ground state depletion imaging. Or you can use photochromic fluorophores. And we saw some of it this morning in Ilaria's talk. And that's called resolved. So, and there are many others as well. So, but they're all based on the same concept. So what do you need to implement this? In principle, it's simple. You need a scanning microscope, like a confocal microscope, and then you need to add a second laser. In some cases, you can use the same one, but generally, you need a second beam, and you have to modulate the phase of that beam, for example, in this way. So this is a very simple way of obtaining this kind of focus. So you see, this is a retardation faceplate, but very particular because it has a retardation that goes like this in a helix. It's called a vortex faceplate. So at each opposite point of this plate, you introduce a phase difference of pi in every opposite point. And then when you focus, so all waves coming at opposite sides arrive at this point with a phase difference of pi, so they kill each other by destructive interference. So at the center, you get a zero of intensity. Half a wavelength to the side, this guy will travel longer and this one less, and then they will arrive again in phase, and they will produce a maximum of intensity, okay? And this happens, of course, with circular symmetry. And you get this kind of focus. This is one way. There are many others. So if you do that, you have all what you need to perform these methods, okay? Let's see a bit more detail how, for example, STED works, this method based on stimulated emission. So you are all familiar with this kind of Jablonski diagrams, right? And this is a simple way to schematically show the fluorescence process. This is the ground electronic state, the excited electronic state, and this is the vibrational modes of each one. And you see that there are several possible transitions to excite the molecule. And that gives rise to the excitation spectrum. Here I'm showing just like 3 transitions to make it easier, okay? Then once the molecule is excited, it will decay to the lowest vibrational level very quickly in picoseconds due to the Boltzmann energy distance of these levels. And from that lower vibrational level of the electronic excited state, the molecule can decay back to the ground state, again to 3 possible levels in this case. There could be less or more. And that gives rise to the detection, to the fluorescence spectrum, okay? So in a typical fluorescence experiment, so you have a fluorophore with an absorption spectrum, an emission spectrum, and what you typically do is, okay, I take some laser light or some lamp, and I take an excitation band somewhere where the molecule is efficiently excited, right? And then I take my detection, I put fluorescence filters or optical filters to collect as much as possible of the fluorescence emission. That's what you typically do. Well, if you want to do STED, you have to change this a bit because you need to sacrifice some of your detection window to introduce your STED beam. So you will reduce your detection window, and here you put another laser. The function of that laser is to bring the molecule down. by stimulated emission. So basically, from the 3 transitions that you had for emission, you will use these 2 to detect fluorescence and this one to bring the molecule back down. Okay, so this laser brings the molecule back to the ground state, and you are not watching in that spectral window, so for you, that molecule is dark. Every time the molecule returns through this channel, the molecule is dark. And that's the way you switch it off. That's the reason why also this changes lifetime, as it was explained in the previous talk. You are competing with the spontaneous decay, so you are increasing the total decay rate, the lifetime is reduced when you do STED. Okay, so here you see some examples. So this really works. I showed you some already. Here you see some more. You can do it in 2 colors, in different ways, by spectral multiplexing, by lifetime multiplexing. Here you see another example from our lab in a trypanosoma cruzi, a system that we study. Again, 2 proteins here, they look more or less uniformly distributed, but when you do super resolution, you see they are organized in clusters that they hardly overlap, but they have many contacts. And then you can start analyzing this in great detail. Okay. We saw already this morning, stimulated emission is not the only way to switch fluorescence off using light. You can also use photochromic molecules or photochromic proteins that you can switch on and off with light, like Ilaria showed this morning. This is called resolved nanoscopy. It's a not very easy acronym. It's reversible saturable optical fluorescence transitions, but resolved sounds nice. And this one, as we saw this morning, it works on the same principle, but uses less intensity of light. Stimulated emission requires high intensities. Stimulated emission is a very unlikely process, so the cross-section is very small. So you need to use very, very high powers to be efficient. Instead, these photochromic transitions are slower, and you can use much milder excitation. And as we saw this morning, this can be parallelized. And that's very important. You can do this with many, many donuts at the same time. And then you only need to scan a unit cell of that pattern and reconstruct the whole, a very large field of view. And here you see some examples. Okay, so I don't need to explain this too much because we had a specialized talk already. Let's move on to the second way. So this is one way, coordinate targeted. Now let's move on to the second way of achieving super resolution. And this is called coordinate stochastic nanoscopy. Now we will switch on and off molecules again, but in a random way, without control of the position, nor the time when the molecules are switched on and off. So how does it work? Again, imagine that you have I explained it in 1 dimension because it's easier like before, but of course this can be extended to more dimensions. So imagine you have, again, fluorescent labels organized in some way, and the distance between them is smaller than the diffraction limit. So if you would register the fluorescence of these labels, if they are all emitting at the same time, you will see something like that. There's no way to tell where each label is, right? So, but now let's imagine again that we have a way to switch molecules on and off. But in this case, randomly. This could be done chemically, physically, we will see later, photochemically. But if we have that, if molecules can switch on and off randomly, we can tune the parameters to get into a regime where most of the molecules are switched off and just some of them are switched on. And those molecules are well separated in space, something like this. Most of them are switched off, and in this case, only these 3 are switched on. So the signal that we will register from each one of them will be diffraction limited. But if we can assure statistically that those signals correspond to one molecule only, even though the signal is broad, we can pinpoint its center very precisely and say, okay, here I have a molecule. Here I have another one, here I have another one. Fine. Now I switch those off and switch on other ones, something like that. And now I can pinpoint the position of these ones. and in a subsequent moment, I pinpoint all the molecules, right? So in this way, I can determine where all my labels are positioned in my sample. So imagine it, now we do it in 2 dimensions. Imagine that you have this smiley face, but the distance between labels is, again, smaller than the diffraction limit. If all the molecules emit at the same time, you get something like that. No way to tell anything, no information. But now we switch most of them off, and we switch these 2 on. So we can determine the position of this one and this one. Next, they are off, and other ones switch on. I can take the positions of this one, and so on. So in this way, I end up not with an image, but with a list of positions. Okay? So the output of this experiment is a list of positions, all the positions of the labels that I could measure, and each one, of course, with its own localization error. So I can then reconstruct an artificial image by putting a dot at the position where I detected each molecule, and with a size of about the error of the determination, right? And in this way, I can reconstruct an image that is no longer limited by diffraction. It's just limited by the localization error of each molecule. And this localization error, of course, depends on the brightness, on the number of photons that I detect from each molecule. So the typical way to localize a molecule, the easiest way, is to use a camera. Basically, this experiment is one uses uniform illumination. So I illuminate the sample uniformly. And then if a molecule switches on, my microscope will make an image of that molecule on a structured detector, like a CCD or a CMOS camera. And that image looks like that. Diffraction limited, of course. But I can make a fit and determine its center. The accuracy with which I will determine the center depends on the number of photons. Here you see, if the molecule is very dim, the error will be high. If the molecule is very bright, I can collect a lot of photons, and I can determine the center much more accurately. Okay? Here you see some example of theory and experiments. Basically, you need about a few thousand photons to reach the 10 nanometer localization error, something like that, in 1 dimension. Okay. So this works. Here you see an example. Imagine, again, you have these microtubules labeled, and if all molecules in the sample emit at the same time, this is what you get, a diffraction limited image. Now, we make them switch on and off randomly, and it looks like that. The sample now looks like this. So we don't get an image, right? We get a video of blinking molecules. We record a video, usually thousands of frames, depending on the size and how many molecules you want to localize, but thousands of frames. And then you analyze the video frame by frame. And in each frame, you make fits of these dots and say, okay, here's a molecule, here's another one, and you start collecting positions. And after that, you reconstruct an image. And now the image looks like that. Resolution is much higher. Okay? Here's a comparison. That's when all molecules emit at the same time. This is when you let them blink and you determine their positions one by one. Okay, so now you know how the 2 families of techniques work, right? So one is switching molecules on and off with light, usually implemented in a scanning microscope. You need to scan that pattern on your sample. This is a wide field technique. You don't need to scan anything. You just illuminate, let the molecules blink, and you can do the same thing. Both families can be extended to multicolor and to 3 dimensions. Okay? In this case, performing 3-dimensional super resolution microscopy is relatively simple. This is one of the most popular approaches. Basically, you introduce a cylindrical lens in your microscope to do astigmatic imaging on purpose. And in that case, if a molecule is in the focal plane, it looks round, symmetric. But if it's displaced in the axial direction, in one direction or in the other, the molecule is elongated on one axis or in the other, just due to the astigmatic imaging. And you can calibrate this, and then every time you detect a molecule, you don't only fit the center, but also the ellipticity, and from that you get the axial position. And it works pretty well. as you can see here. You see bundles of microtubules going on top of the other and stuff like that. Okay. So these methods are really, really great. Here you see another example. You can do it. This is from the lab of Christophe Leterrier. And you can do wide fields, get very high resolution in 3 dimensions, and you can explore different areas. This is, again, this membrane-associated periodic skeleton of neurons. You can do multicolor, of course, either spectrally or based on other methods. Here you see, again, the same parasite I showed you before instead, but now we will analyze it with the single molecule localization. 2 proteins that we are interested in. This, I mean, the particular details doesn't matter, but just to give you an idea, this is a parasite that produces Chagas disease. It's an endemic disease in South America and North America as well. And this is a parasite that survives in the host. It evades the immune system of the host by covering its membrane with sialic acid. But it cannot produce sialic acid, so it has to capture it from the bloodstream of the host and put it somehow on the membrane. So for that purpose, it has a specific enzyme. It's called transsialidases. And this is the enzyme that captures sialic acid, and it stores and accumulates sialic acid in other proteins that are mucins. So we labeled both of them to take a look at how they are distributed in the membrane. On a diffraction-limited fluorescence microscope, it looks like that. There's no way to tell much of what's going on. Now you let them blink in 2 colors, and you reconstruct the positions of the proteins, and you get this kind of image. And you see again, these proteins are organized in clusters, and now you can start measuring cluster sizes, distances between them, compare them to random distributions of proteins, and trying to learn some about the biology of this process. Okay. Again, as in the case of coordinate targeted, in coordinate stochastic super resolution, there are many methods possible. And the difference between them is the mechanism to make the molecules switch on and off randomly. Okay? In this case, it doesn't have to be induced by light. Could be, but it's not necessary. It has to be random. So one way is like using photochromic fluorophores. In this case, it was induced by light, but on a wide field, so it has a cross-section, it's stochastic, some molecules were switching on and off randomly. You could use the excursions to the triplet state, of course. This is also a rather universal mechanism. And again, those excursions are random. So if you can tune the triplet state to be long enough, you can manage to put most of the molecules off, and then some of them will recover randomly and blink, and will let you do the method that I just described. But you can also use physical mechanisms like adsorption. This is a very simple thing. So in this case, what people did was just let dyes adsorb on a surface. And if this adsorption time can be tuned, the molecules that are diffusing in solution are just too fast and they make a uniform background. But when a molecule adsorbs, then it remains there on a fixed position for some hundreds of milliseconds, and you can make a good image, and then you can pinpoint its position. So this is just blinking by adsorption. An improvement of this is the technique called DNA paint, in which the structure that you want to image is labeled now not with a fluorophore, but with an oligonucleotide, a short single-stranded DNA chain. And then you put in solution the complementary oligonucleotide. This one is labeled, right? And it's designed to hybridize here only transiently. So it will hybridize for some time, and then it will go back to the solution. So usually you use about between 7 and 9 complementary bases. And then the binding energy is comparable to KT. So the binding lasts between some milliseconds to 1 second, and it goes away. And then the next time, here or in the same position or in another, randomly, they will hybridize and give you a bright spot from which you can pinpoint the positions of your sample that you labeled. Okay? Here's an experiment. This is an artificial structure. It is a DNA origami with 3 docking sites for DNA, separated by 80 nanometers each, subdiffraction distances. Tomorrow I will give another lecture. I will explain a bit about DNA origami. But for today, you just think that we have some artificial structure with 3 docking strands for DNA at sub-diffraction distances. And we let them blink. And then we analyze this video and reconstruct an image. And this is what you get. A lot of points, but all of them are in one line, and there are 3 dots and separated by the design distance of this DNA origami. So you see, you can really resolve sub-diffraction positions in a sample. And this can be, of course, used also in biological samples. So all you need to do is to label your target molecule using an antibody, an antibody, or any other labeling strategy. Any labeling strategy that you used to put the fluorophore on your target protein, well, now you have to put an oligonucleotide. That's the only difference. And then you put in your sample the imager strands, and they will bind transiently and reveal the position of that protein. An interesting feature of this is the possibility of sequential multiplexing, because in this case, for example, we want to do 2 colors, just 2. What do? You put one DNA sequence on one of the proteins and a different DNA sequence in the other protein. So you first incubate your sample with an imager strand that is complementary to one protein. And then you will see blinking at the positions with that DNA sequence. Once you have collected enough positions of that protein, you rinse and you inject the next imager with the other DNA sequence, right? And this one will bind to the other protein. And now you again measure until you collect enough positions of that second protein. And after that, you can reconstruct a 2-color image, okay? With 1 color microscope. So here you don't have to worry about aligning 2 color channels, about chromatic aberrations or anything like that. You just have to be patient because this is sequential. You measure one, then the other, then the other. But if you have a stabilized microscope, you can do this many, many times. And here you see a rather extreme example from the lab of Ralph Jungmann, where they measured 30 different proteins in a neuron with super resolution, just by sequential multiplexing of DNA paint. And in this way, okay, in this case, here the color scheme is not very clear, but here you see the 30 different proteins they measured in super resolution. And now you can do this as a spatial proteomics. Like you can see how proteins are organized in space with nanometric resolution and try to identify even previously unknown interactions between proteins. Okay. So that brings me to an end, and let's make just a summary. So we saw 2 ways of breaking the diffraction limits. The first one is called coordinate targeted, and it is based on light-driven suppression of fluorescence. And for that, you need a structured illumination. That's the key point. You need to control the region of space where you excite and where you suppress fluorescence. Typically, you need to leave holes in your pattern where you don't suppress, right? Like using this donut or multiple donuts or interference patterns like we saw. But you need to do that. So in this case, in this kind of methods, all the information to achieve super resolution is based on the illumination. That's what you need to learn. After that, you just use a point detector, a photomultiplier, and a photodiode. So you don't use any information on the detection side. All the information is here on the excitation. The other family is kind of the opposite. The other family is the coordinate stochastic family, where you let molecules blink, and every time they blink, you make an image on a structure detector, and you pinpoint its position. So in this kind of experiments, you don't inject any information with the illumination. Your illumination is actually uniform. So you want it to be uniform so that all molecules are excited the same way. They are all equal in terms of intensity and brightness, etc. So, and then all the information becomes from the detection, because you will make a fit to the signal and pinpoint its position. So you see, they are kind of complementary. In both cases, achieving high resolution requires a large number of fluorescence photons, okay? So these methods have no fundamental limits. So in principle, they could reach any resolution that you want. Theoretically, they have no limit. In practice, they do have a limit because you need a lot of photons. For example, here, in principle, just collecting one fluorescence photon could tell you there is a molecule here. Because imagine, you could do an experiment, you switch on these two lasers, you wait for a while, if you collect a photon, you say, okay, here's a molecule. If you wait some time and you didn't collect any photon, so with some statistical criteria, you could say there's no molecule, and you move to the next position. You wait again for some time. But in practice, this is not possible. Why? Because there's always background. So you always collect some background signal. So you need fluorescent signal that has to be significantly above background to make this kind of to address this kind of question, whether there is a molecule or not. So at the end of the day, you need a lot of fluorescence photons to achieve high resolution. And in this case, it's even more direct because the precision of the localization algorithm depends inversely with the square root of the number of photons directly. So if you want to have a low localization error, you need a lot of photons. In both cases, you need a lot of photons. And that is the reason why, even though these methods have no fundamental limit, they face a practical limit. And that practical limit is of about a few tenths of nanometers in the lateral dimension and 5 to 3 times worse in the axial direction. So something like you can see here. So it's wonderful. You can discover things, as we saw. But let's imagine, for example, here we see these microtubules much better than in a diffraction limited image. We can clearly start studying microtubule organization, no doubt, and here it's impossible. But what if we would like to see molecular organization? So microtubules, we know, at least from crystallographic studies, that they are, so if we made zoom, the image gets blurred. But we know that this should be a quasi-helicoidal structure of alpha and beta tubulin, right? But we cannot see that here. Why? Well, one of the reasons is because we don't have enough resolution. So the distance between tubulins is about 8 nanometers. But our resolution is 2 or 3 or 4 times that. So there's no way to see that. So we need the next push in the resolution to surpass the 10 nanometer regime. So this is not because it's a round number, more or less, but it's also because structural proteins have a size of about 10 nanometers. So if we have a better resolution, we can start seeing how individual proteins are organized to form these supramolecular structures that then give rise to the subcellular structures, cells, and tissues, and organs, etc. To really look at this at the molecular level. So to do that, we need some new tricks. Fundamentally, actually, there's no limit, but there is a limit in practice. So that gave rise to what I call like a second generation of super resolution methods that can solve that limit. And tomorrow at 9 o'clock is the second chapter, and we will see some of those methods. Thank you very much.
Thank you very much, Fernando. So now we have time for some questions. Fernando?
Yes.
Thank you. Very nice talk. Can I ask you in Spanish?
Yes.
Perdón. Vos dijiste que en las técnicas como Storm uno suele usar wide field. Entonces, ¿cómo vos vas a tener un montón de blinking que va a estar a diferentes planos y que te van a generar eso, me imagino, mucho más error en las localizaciones? Te quería preguntar qué pensás de si uno conociera la PCF de su microscopio, uno podría hacer un procesamiento de tal manera de eliminar el blinking que uno por ahí por el diámetro que tiene considera que está en otro plano.
Okay, very good question. We'll just repeat it in English. Yes. So the question is about what happens in single molecule localization microscopy, like STORM or PAINT, these methods that work on blinking molecules, what happens to the molecules that are out of plane? Because it is true, I was explaining everything here in 1 dimension or in 2 dimension. I was neglecting the other dimension here. But all these concepts, I showed you some of the 3D, but in principle, these concepts can be extended to 3D. But if you don't, so if you just take a conventional microscope and you don't do any trick, and like here, for example, or here in all these examples, what you will collect is a projection of all the optical section that you are analyzing. So basically your illumination, let me come to this slide. Yes, okay. So you use wide field illumination. So in the illumination, there's no information. Everything is the same. Now you make an image with a microscope. When you make an image, you are selecting a focal plane. That focal plane is diffraction limited. So basically, you're imaging on your camera, let's say, a plane that has a width of about 500, 600 nanometers. That's where your molecules will look bright and round. If they are out of that plane, they will look out of focus. So normally, when you analyze your data, right, you get signals. You register this kind of signal, right? You're looking at one plane. If a molecule is out of plane, it will look broader. When you analyze these frames, frame by frame, you will choose one of these dots and make a fit. And then you apply a lot of filters in your fit. For example, one of the filters could be that it should be round. If it's not, it doesn't meet some criteria of symmetry, you could say here that maybe there were 2 molecules. I don't want that. Another criteria could be brightness, because they should be all equally bright. So if something is too bright, you discard it, because it's maybe 2 or more molecules. Another criteria is the size. So out of focus molecules are usually discarded during this post-processing, either because they are too dim, because the signals are too dim, or the signals are too broad. Okay? So at the end of the day, you collect an image that is a projection of all those 500 to 600 nanometers. Okay? That is if you don't do anything to achieve optical sectioning. But you can do something. For example, this. So if you apply astigmatic imaging, now you're collecting a signal from those 500 to 600 nanometers. But you see that the molecules are the signals are slightly distorted. Now, when you analyze this, you will not apply any filter for asymmetry, because you want to pay attention to the asymmetry. And that asymmetry will give you the axial position. Some people use more sophisticated PSF's algorithm, more distortions. Oh, so, was it me? Okay. Like helicoidal PSFs or any other things to encode the axial position. Okay, but if you don't do anything like of that, you are projecting all that